Hello, everybody, and welcome back to BioSC 140, Human Physiology. This video is for Lab 11 and 12, Frog Nerve and Muscle Physiology discussion. So let's review real quickly what the experiment is. We're going to be taking out a frog sciatic nerve. We're going to be shocking one end of that frog sciatic nerve and measuring how much electricity, measuring the response on the other side of that frog sciatic nerve. We're going to be shocking the nerve with varying amounts of electricity and seeing how varying the amount of electricity varies the amount of response. With the muscle, we're going to do the same similar procedure where we shock the gastrocnemius of a frog with varying levels of intensity of electricity. And we're going to be measuring how much force is generated by the contraction of the muscle. Once we have found what voltage elicits the maximum amount of force generated, we're going to be doing something that we didn't do with the nerve. We're going to see how frequency affects force generated. So we're going to start to shock the skeletal muscle of the gastroc of the frog once a second for a few seconds. See how that changes the amount of force generated. Twice a second, then 4 times a second, 6 times a second, 8 times a second, 10 times a second, and so on. And seeing how frequency affects the amount of force generated. So some background information. The frog sciatic nerve, so I want to make a distinction. A nerve refers to a bundle of peripheral nervous system axons. PNS is peripheral nervous system axons. A neuron is a single nerve cell. So when you talk about the sciatic nerve, it's a bundle of peripheral nervous system axons. When you talk about a neuron, it's a single nerve cell. Now, we have 2 main types of peripheral nervous system cells. We have afferent cells, which bring information from sensors to our central nervous system. And then we have motor or efferent fibers, which send signals from our central nervous system out to something that can affect change. The frog sciatic nerve is a mixed nerve, so it has both afferent and efferent fibers. It has around 1,500 of these nerve cell axons. The frog nerve cell is going to, a cross-section of the sciatic nerve in the frog is going to look something like this. You're going to have bunches, you're going to have basically a big bundle of nerve axons. You can see all these little circles are different frog cell axons. Here's another image of a cross-section of a nerve. All those little yellow dots, if you zoom in, are different axons. So it's like a rope. A rope has many strands. There's many axons within this mixed nerve, the sciatic nerve. So, action potential. So I'm going to be repeating myself a little bit in this video because I need to make some clear distinctions that students often get mixed up on. So an action potential. An action potential is when a nerve cell sends a signal down its axon. So the nervous system is famous for sending electrical signals. They send those electrical signals down something called an axon, which is like a long arm coming off the cell. This next part's really important. Every single time a action potential happens, a signal is sent down that axon, the electrical reading looks something like this. And every single time, the amplitude of that change is the exact same. I'm going to repeat this. You can see right here. Every single time an action potential happens on an individual axon, the amount of electricity, the intensity of the electricity is the exact same. Exact same. And it's the exact same at any point along that fiber. So at the beginning of the fiber, exact same. Middle of the fiber, exact same. End of the fiber, exact same. Anytime there's an action potential, it always has the same amplitude. You can see a resting potential of the cell is like negative 70 millivolts. Once that nerve cell depolarizes to about negative 55 millivolts, the action potential is going to happen. And it's going to go from negative 55 to about positive 30, and then back down to that resting state. Every time an individual axon, every time there's an individual nerve cell does an action potential, it's this amplitude every single time. What causes that individual cell to do an action potential? Well, the cell, which is at a resting state of negative 70 millivolts, depolarizes, or the charge across the membrane of that cell changes to negative 55. At that point, the action potential is going to occur. At that point, the action potential is going to occur every single time at the same intensity. An individual action potential of an individual nerve is said to be all or none. You don't get a half an action potential. It's impossible. You either get a full action potential or you get no action potential. The nerves either sends that signal down the axon or it doesn't. It can't send a little one, it can't send a big one. It either sends the action potential, same amplitude every time, or it doesn't. It's considered all or nothing. You get the whole thing or you get nothing, all or nothing for an individual action potential. Individual one fiber action potential is all or none. You either get it or you don't. If it's above threshold, threshold is the change in membrane potential. If the cell depolarizes, if the charge across the membrane changes to negative 55 or higher, so negative 54, negative 53, the action potential will travel with a constant amplitude the whole way down the axon. Constant amplitude the whole way down the axon. Now, once we get into the nerve lecture, we're going to know exactly why and how this happens this way. For right now, you kind of just have to trust me on this because you need to know this for this lab. CAP stands for compound action potential. Compound action potential. And a compound action potential is when you look at a group of neurons, a group of axons. When you have lots of different axons, or 1,500 in the case of the frog nerve, you have a compound action potential. Let me take a step back. You have the potential for a compound action potential when you have a bundle of these axons together. A compound action potential is the combination of individual fiber action potentials. So we just talked about the characteristics of an individual action potential. Right now is when you're combining multiple axons, the combination of individual fiber action potentials. Individual APs are all or nothing. They always have the same intensity. You either get them or you don't. Compound action potentials are considered graded. So what we're going to do is in this lab is imagine this is our sciatic nerve right here. It's going to be resting on an electrode right below it. As we increase the strength of the stimulus, the strength of the shock that we're delivering to the sciatic nerve, we're going to hit a point where the closest cells to that electrode depolarize to -55, which means they've hit that threshold value, which means they're going to send their action potential. We're then going to, but at the first one, only the cells, only the axons closest to the electrode are going to send those messages. As we increase, so only in the area where my cursor is right now, as we increase the voltage, we're going to be increasing the number of fibers sending that signal. As we increase our voltage further, we're going to increase the number of axons sending that signal. And then we increase the voltage again. We're going to increase, and we're going to increase, and we're going to increase until every time we shock that sciatic nerve, every single axon is sending the signal. That's a compound action potential. How you can have more or fewer axons sending that signal at a time. This is considered graded. Graded is like different amounts of output. So you can have different amounts of output, different amounts of electrical signal being transported because you have a different number of axons sending signals. Stronger the stimulus, the stronger the response. Know the difference, be able to explain the difference between compound action potentials and individual fibers. So compound action potential, it's like having a varying number of lights on. Each light is an individual action potential. You can combine a bunch of action potentials to get a larger output. So we're going to be shocking the sciatic nerve with varying amounts of stimulus. And once we get to the point where the cells closest to the pin reach their threshold value, -55, we're going to start seeing a response. We're going to start seeing this action potential, this compound action potential response. This initial shock you see right here is an artifact from the stimulus. And then you can see this nice smooth response wave right there for the action potential. We're going to be measuring the low point and the high point of that action potential response. And we're going to get this amplitude of our action potential for different stimulus for voltages. So what we're going to notice is that at the lowest voltages, not a single one of these nerve fibers is going to send an action potential. They will not reach threshold. They will be below the threshold required to cause any action potentials to occur. They will be called subthreshold and will get no response through the sciatic nerve. As we increase the voltage, we're going to reach threshold. Threshold is the smallest voltage that elicits a response. So it's the smallest voltage that gets the smallest number, the first voltage that gets a response, and it's going to activate, it's going to get a response from just a handful of those axons. We're then going to increase the voltage, and we're going to, you see, increase the amount of response we get at the end. At some point, increasing the amount of shock is not going to increase the amount of response that we get. You can see anything greater than 0.9, we got the exact same response as 0.9. 0.9 is, or the maximal voltage, is the voltage above which no increase in response occurs. So once we hit a voltage where no increase in signal sent occurs, we're going to call that the maximal voltage, that lowest value that gets the max amount of signal sent. What's happening physiologically at that maximal point is every single axon is reaching threshold. The maximal voltage is the voltage required to get every single axon within the sciatic nerve to send that signal. And anything above it, you're still going to get every axon sending the signal. And remember, every signal that is sent by an individual action potential, it sends the same intensity signal every time. Same intensity signal every time. So really your maximum is the intensity of that individual signal times 1500 for each of the fibers within that compound action potential within that uh, frog sciatic nerve. So there is a point where you cannot send any more signal, the maximum. Anything above the maximal is the supramaximal. So subthreshold, no fibers. We shock the nerve, no fibers reach action potential, no fibers send an action potential. Threshold is the smallest charge that gets some response, that gets some fibers to reach action potential. Submaximal is more stimulation, more fibers. So as we increase the voltage, we're going to increase the number of action potentials, of fibers sending action potentials. The maximal is the minimum voltage required to get a response from every single axon within the nerve. Supramaximal is above that. We've mentioned this before in class. This is demonstrating spatial summation. What we're trying to demonstrate here is spatial summation. We can't increase the intensity of an individual action potential because the intensity of an individual action potential is the same every time. So if we want to send a stronger signal, we have more fibers sending a message at the same time. Spatial summation. Increased response due to increased recruitment of active units or fibers. More stimulus, more fibers, more response. Single separate stimulation events means only spatial summation was observed. So with the frog sciatic nerve, we were only investigating spatial summation because when we shock the nerve, we shock it for a moment and stop. Shock it for a moment and stop. There's no temporal summation that comes into play with the frog sciatic nerve. After all of us are done with the frog sciatic nerve experiment and all of our data has been gathered, I go around and I drop lidocaine, which is a really common anesthetic, on each of our nerves. And then we shock the nerve again. And what we find is that there's no signal sent. There's no signal sent when we drop the lidocaine on the nerve. And that's because lidocaine blocks sodium channels. And we're back to the sodium-potassium pump, maintaining that membrane potential. When sodium can't pass through the cell membrane while those channels are blocked, it blocks action potentials from happening. Lidocaine, it prevents pain signals in low doses. Muscle. So muscles are actually kind of similar in arrangement in the sense that they're bundles of individual fibers. Bundles of individual fibers. And each muscle fiber is a contractile unit. Now, the phase one of this, part one of the frog gastrocnemius experiment is similar to the sciatic nerve experiment in the sense that we are shocking the gastrocnemius with varying levels of electricity, and we're seeing the response. Each shock has tons of time between them. It's like an individual shock, and then we wait, and an individual shock. So we're going to be looking at spatial summation in this first part of the frog muscle lab. Muscles are also similar to nerves in the sense that when they're shocked by electricity, they can have what's called a muscle action potential, where when you have a muscle action potential, you're going to have a change within the muscle cell. where the charge changes on the muscle cell, and it's going to open up channels on something called a sarcoplasmic reticulum. It's basically this little organelle within muscle cells which holds calcium. And when that organelle opens up and releases calcium into the rest of the cell, it's going to start the muscle contraction. So muscle action potential. When we first start shocking the muscle, we're not going to get any response because the amount of force that we start shocking the muscle with is going to be below threshold. It's going to be sub-threshold. And as we increase the amount of force used, we're going to cause more and more and more muscle fibers to reach action potential. And when we have more and more muscle fibers reaching action potential, we're going to be seeing spatial summation. We're going to be seeing an increase in force generated. So if you look here, this is what the readout looks like in lab. This readout up here shows the stimulus, the amount of shock that we're shocking the muscle each time. So shock here, shock here, shock here. Down here, you can see the muscle response in grams, the amount of tension that's produced by the frog muscle. You can see down here at the smallest shocks, there's no muscle tension produced. It is sub-threshold. It does not cause any muscles to reach action potential these first two shocks. On the third shock, we start to get some response. That's the threshold voltage. So procedure increased strength, perfect. So right here, you can see below 0.4, no tension, sub-threshold. 0.4 is the first voltage where we get tension, threshold. As we increase voltage, we get more and more force. And at some point, in this example, 1.2 volts, we're going to reach our maximal response. Maximal response is the voltage at which, if you increase it, there's no increase in force generated. And that's because every muscle fiber is being recruited. Every muscle fiber is having a muscle action potential at this point. Every muscle is contracting at this point. Subthreshold, no fibers. Threshold, above this, we're getting some muscle action potentials. Maximal, all fibers, and you can't increase beyond all fibers contracted. Spatial summation. Spatial summation, increased response due to increased recruitment of active units. So a muscle twitch. A few things before we move on to temporal summation with the muscle fiber, with the muscle. A twitch is a single muscle response to a single stimulus. This is what we've seen so far. It's when you have an above threshold stimulus and it's a single stimulus and the muscle can completely relax in between each stimulus. So let's look at this for a moment. There's going to be three distinct phases to a muscle twitch. There's the latent period. So if you look here, we shock the muscle, and there's a period of time before the muscle contracts. This is the time period. There's a number of things going on, but the main thing that you should know about is this is the time it takes for that calcium to flow out of the sarcoplasmic reticulum and into the cytoplasm of the cell. When that calcium flows out, it starts the process of a muscle contraction. Once that calcium floods out and it starts allowing cross-bridging, we're going to start getting the contraction phase. We're going to start getting contraction within the muscle. With that stimulus, with it being just a quick, short stimulus, what we're going to start to see is that after a short period of time, all that calcium is going to start getting actively transported back into the sarcoplasmic reticulum. And as calcium levels start to decrease in the muscle cell, we're going to start seeing a decrease in the amount of force generated. So contraction phase, relaxation phase is when the tension is decreasing, all that calcium is being put back into the sarcoplasmic reticulum. And this happens really, really fast. You can see, milliseconds time, really fast. So latency period is the lag from stimulation to contraction. Contraction is the beginning of tension to the peak, and relaxation is the peak back to the baseline. So you can see right here at the latency period, we have the nerve sending the signal, the action potential to the muscle, and it releases neurotransmitters, so chemical signals that are going to cause the muscle action potential to occur. When the muscle depolarizes enough for an action potential to occur, it's going to cause this sarcoplasmic reticulum to release calcium. When calcium is released in the cytoplasm, it's going to cause the muscle fiber, the actin and myosin filaments, to form cross bridges, which is what generates the force within the muscle cell. During the relaxation phase, calcium is going to be resequestered back into the sarcoplasmic reticulum, which is going to prevent the actin and myosin from interacting with each other. And so we get less tension to no tension produced. Relaxation phase. More calcium, more cross-bridging, more interaction between actin and myosin, more tension. So what we're going to do in this last part of this lab is we're going to see how changing the frequency of shocks to the muscle is going to change the amount of force produced. So we're going to increase the frequency of a maximum stimulus. We're using a maximum stimulus because we already know that at maximum stimulus, every muscle is being recruited. So spatial summation is not a possibility to be the reason for increasing the force generated by the muscle. The increase has to be due to the frequency because every muscle fiber is already being recruited. It can't be due to the strength of the signal or because of recruiting more signals, more fibers. So what we're going to see is that there's going to be four distinct kind of results that we see, four distinct types of these contractions. So twitch is what we saw in the initial part one of the muscle lab, where we have the contraction and then there's a complete return to baseline. So at about 1 hertz or about 1 shock a second, we're going to see no summation, meaning no temporal summation. We're going to have a complete return to baseline in between each muscle action potential. There's going to be no temporal summation, Complete relaxation. All the calcium that's released in the cytoplasm is going to be transported back into the sarcoplasmic reticulum. During TREP, which is a German word which means staircase, it's 2 hertz to 6 hertz, so 2 shocks a second to 6 shocks a second. We're going to start to see a little bit of summation of force. So a little bit increase in force, 2 to 6, right here. A little bit increase in force, and then not a complete return to baseline, a complete return to baseline with a little increase in force. So a little increase in force, but complete return to baseline during relaxation. And we're going to see this because not all the calcium that's released during the initial shock, during the initial muscle action potential, is going to be able to be transported back into the sarcoplasmic reticulum before another muscle action occurs. So some of that calcium is going to stay in the cytoplasm and not get moved back into the sarcoplasmic reticulum before the next shock occurs. But enough calcium is going to get put back into the sarcoplasmic reticulum so that the muscle will relax back to a baseline relaxation. Once we get up to 6 to 10 hertz, we're going to start seeing something called wave summation. We're going to continue to see an increase in the amount of force, and we're not going to see a complete return to baseline. So the amount of force generated is never going to return to 0. It will always be above 0 during wave summation. This is going to be due to a continuing increased amount of calcium in the cytoplasm. So even less calcium is getting transported back into the sarcoplasmic reticulum between shocks. The calcium is going to stay in the cytoplasm and continue to allow actin and myosin to interact and form cross bridges and produce force. Another reason why we're going to see this no return to baseline is because the pre-stretching of series elastic elements. So there's a stretchy component to our muscle cells. And that stretchiness, it's going to be at maximum stretch the entire time. With twitch and trep, we're not going to be at maximum stretch the whole time. There's going to be some relaxation, some decrease in the amount of stretch within the muscle cell. Once we get to wave summation, there's no decrease in the amount of stress. It's always maximum stretch. At tetanus, we're going to see the highest amount of summation, the highest amount of force generated. There's going to be no relaxation at all. All that calcium is going to stay in the cytoplasm all the time. The amount of calcium getting transported back into the sarcoplasmic reticulum is negligent. Further increasing calcium concentration within the muscle cell, pre-stretching series elastic elements. So there's always going to be full stretch on our muscle cells. Tonic muscle recruitment. So in humans, we only have tonic muscles in a few places in a few specific examples. With the frogs have these tonic muscle fibers within their gastroc, again, rare in humans, but they're going to respond. They're not really going to have action potentials with just stimulus, with just a single shock. They activate once they get a high temporal frequency of shock. So that's going to be another reason why we get more contraction with the tetanus. So we're observing temporal summation, increased response due to increased frequency of active units firing. Maximum stimulation means only temporal summation was observed when we're increasing the frequency. So part one of the muscle, we demonstrated spatial summation. Varying responses recruits varying amounts of muscle fibers to contract. Part two, we're experiencing temporal summation. All right, if you have any questions, please reach out and have a great day.